Hello and welcome to Better Biopharma, the official podcast of Bioprocess Online. I'm your host, Tyler Menichiello, and today I'm happy to be joined by Paul Romness and Dr. Robert Petit from OS Therapies, where Paul is the chair, CEO, and president, and Robert is the chief medical and scientific officer, as well as the founding scientist behind the company's lead asset, OST HER2, immunotherapy for osteosarcoma and other HER2-positive cancers. Paul and Robert, thank you both so much for being here.
Pleasure.
Pleasure.
So to kick us off, Dr. Petit, would you mind explaining the company's lead candidate, OST HER2, to our audience?
Certainly. We've known for a number of years that the best defense the body has against cancer is the generation of a white blood cell called a T cell. OST HER2 uses a very creative system. It uses a live bacterial vector that's been genetically modified to attenuate it or change it so it will not cause disease. But once it gets the attention of the immune system, it secretes tumor antigen targets. And then the immune system sees that in association with this potential pathogen and generates a T cell response to try to fight against what may be a pathogen infection. And what it finds is that the cancer cells are expressing these targets. And so the immune system ends up hunting down cancer cells throughout the body and killing them wherever they are. It's a fascinating use of a live bacterial vector that gets the attention of the immune system in a different way than what the cancer has, because the cancer is being tolerated, but the body, even if it has cancer, will still fight off an infection. So we're leveraging that aspect of the immune system to sort of trick the immune system into going after the cancer cells, seeing them in a different way.
Yeah, thank you. The Listeria-based bacterial vector caught my attention because you don't see that every day, a living vector. And from Paul or Robert, either one of you, what kind of unique considerations come with using Listeria-based bacterial vector? Or maybe taking a step back, what is behind that decision? And what are some unique considerations when it comes to using a bacterial vector?
Well, I'll start first. Listeria has a very unique lifestyle amongst any of the bacteria, in that when it gets inside of any mammal, whether it's a dog or a human or whatever, it gets gobbled up by the immune system's collectors, the phagocytic cells. And normally that's the end of a bacteria. But Listeria has a little trick. Once it finds itself gobbled up by the antigen presenting cells, it then escapes into the central part of that immune cell and spends the rest of its life inside of other cells. And so antibodies and things in the bloodstream are not able to get access to Listeria in a wild type infection. So what that means is that the body has figured out that, hey, there's no point in making antibodies to this thing. It's hiding inside of cells where we can't get to it. So it generates a T cell specific immunity. And that's the same type of immunity that we use to kill cancer cells. So it takes a T cell to kill a potentially infected cell, that takes a T cell to kill a cancer cell. And so that's a really unique aspect of it. But unlike a virus that can be neutralized by antibodies, Listeria vectors are not neutralized by antibodies because they hide inside the cells. And so you can give this over and over and over again, repeatedly boosting the immune response so that the immune system just gets stronger and stronger and stronger against the targets that the Listeria is presenting. That's one unique aspect. The other unique aspect is it's still a bacteria. And as a bacteria, it's completely eliminated by simple antibiotics like penicillin or Bactrim. And so once we essentially kind of use the Listeria as a UPS truck to deliver the package of target information to the immune system. And once the package has been delivered, in our case, it's about 4 hours later, we simply give antibiotics and then the UPS trucks blow up. There's no need for them, and they're eliminated by the antibiotics. And so there's no persistence of the vector. And this is really a wonderful, unique aspect of using Listeria as a vector as opposed to a virus, which you can only use once or twice before neutralizing antibodies make it ineffective.
What a visual of a combustive vector.
UPS may not like that analogy. I didn't.
And Paul, I'm curious maybe to hear from you on the operational, maybe CMC challenges as it pertains to developing OST HER2. When it comes to developing a Listeria-based vector, what CMC challenges would you say there are, especially in terms of formulation and safety? I imagine there's some special considerations there.
Well, I'm happy to have Robert get into the details of it, but, we are dealing with a live attenuated, Listeria with a HER2 vector fused to it, and luckily it's not as potent as a typical Listeria, which has massive immune responses, but the immune response is equal because the body's reading it as, full-strength Listeria. From a CMC standpoint, that's very timely, Tyler, because we're putting together the CMC section of our biologic licensing application for the FDA, and luckily we've got about over 90% of the CMC section done. But I'll let Robert comment more on the specifics around dealing with Listeria, especially in manufacturing and storage.
Yeah, so there are a few interesting aspects of it. One, the product itself is a live bacteria, so you cannot really use a sterile filter to infuse it through because then you'll be eliminating the drug. And so, from the manufacturing perspective, what we do is we confirm that the mutations that make it unable to cause disease are still in place. Those deletion mutations are in fact still deleted. We also confirm that it makes the target products, the target tumor antigens that we needed to. That's secreted in the as part of the manufacturing control. But once the genetic manipulation is initially conducted, where we delete the genes of interest or of that cause disease, and we insert extra chromosomal DNA that codes for the secretion of, in this case, the HER2 antigen targets. We just confirm that happens, and then the manufacturing process becomes very simple. It's just a simple bacterial fermentation, which is the industry is very common with, because a lot of drugs are produced in E. coli and other things that are fermented, but then they purify the drug from the liquid. This is not the case here. We're not purifying any drugs from the liquid around the bacteria. The bacteria themselves are the drug. And so it's, in some ways, it's a little bit simpler than other more biochemical manufacturing processes.
Yeah. What you said, that was really interesting about how you can't be sterile or aseptic when their final product is literally a bacterium. Let's dive into that a little bit. I'm curious to hear, how do you maintain that the only bacteria present is the bacteria that you want? Is there any level of maybe genetic engineering or antibiotic resistance, or how do you select for?
Well, there's a lot of genetic characterization that goes on. What we do is we ensure monosepsis, means there's only one bacteria there. So we screen for all sorts of other extraneous bacterial DNA and viral DNA or any other potential contamination, and make sure none of that is present. And then we have only the bacteria of interest, and then we sequence the DNA of the strain and make certain that it's not wild type Listeria, which would really mess us up, but it's our specific weakened. You can say that it's kind of like a pet bacteria. If a wolf is an animal that'll eat you, a dog is your best friend. And so, in a way, wild type Listeria can and does potentially cause disease. But after we domesticate it and take out the genes that allow it to move around and cause disease, it becomes a friendly dog that basically is delivering the payload that we need. So, monosepsis we ensure, and we make sure there's no other adventitious bacteria or viruses, and that involves a lot of culturing and a lot of sequencing of any extraneous DNA to make sure that there's nothing in there that we don't intend to be there.
Yeah. Thank you. That makes sense. What other manufacturing differences are there? Like, how does the process to manufacture a live attenuated bacterium vaccine differ from traditional vector manufacturing even?
Yeah, one of the most interesting things about this is these bacteria are cultivated in a closed system. You know, it's they have plastic bags that are on rockers that gently rock it back and forth, and it's gas permeable bags, so they can get the CO2 and oxygen exchange that they need. But it's a closed system, and we inoculate it with the Listeria, so there's no opportunity for the Listeria to get out or other bacteria to get in. And so that's really fascinating, because as opposed to putting a giant, 500-liter stainless steel vessel of E. coli or whatever, this is all done in a closed system. And when it's cultured properly and it's doing really well, we can get 1 times 10 to the 10th bacteria or Listeria vectors per milliliter of the culture. And so that's 10 doses of the drug for every milliliter of culture. So it doesn't take a giant volume either. So it's very efficient to manufacture, and the manufacturing controls are just about ensuring monosepsis and that it is what that we identify it to be. It's carrying the payload and functions properly, and is not carrying anything that's not supposed to be there.
Yeah. Thank you, Robert. Paul, I had a question for you on the manufacturing patent regarding um, your lead candidate here, OST-HR2. I read that OS Therapies extended the manufacturing patent exclusivity to 2040. I was curious, how's the company thinking about IP protection, and how does that affect working with partners, maybe in whether it's scaling capacity or even your approach to tech transfers? How do you contend with all that? I'm curious to hear your thoughts.
So that patent extension for lyophilization process is very helpful. We recently acquired the entire platform, Listeria platform, not just HER2. When we started OS Therapies, before Robert even came to join his technology, uh, we only had osteosarcoma as an indication for humans. And then we added the indication for canines, which is a big deal. 30 to 40,000 dogs a year get osteosarcoma. Then we added the other indications for the HER2, but there was prostate and some other technologies based with the Listeria that we didn't have the entire platform. Since we acquired that earlier this year, and then adding that time to the IP to 2040 really gives us the opportunity to use this platform the way it should be, as a platform technology. There is a platform designation with the FDA that we'll be going for. And we see many other indications beyond osteosarcoma. So partnering with some potential bigger partners, it's really helpful to have this increased IP to 2040
Yeah. Thank
on
you,
the whole
Paul.
platform.
Thank you. Um, and with the phase 2B trial complete and BLA on the horizon later this year, how is the company preparing manufacturing operations for commercial scale?
So Gerald or Robert can get into the details of the manufacturing, but we do use contract manufacturing, and it's the same manufacturing probably that manufactured the original clinical trial drug. Just very high quality, which is most important. And then that lyophilization will occur with that same organization.
Yeah. And I was curious, either one of you wants to weigh in here on how this manufacturing process for OST HER2 was any level of technology borrowed from another notable therapy on the market or in development, or is this kind of bacterial vector approach super unique to OS?
Yeah. So
You were there in the beginning.
Yes. Thanks, Tyler. Yeah, HER2 is clearly a validated target for cancer immunity. There are HER2 blocking drugs where HER2 is a tumor driver that have been approved several generations. There are even they even use a targeted, uh, you know, a targeted form of chemotherapy called an ADC that targets against HER2. So HER2 is definitely a validated anti-cancer target and has been um, eh, shown to be effective, across a couple of different technology platforms. And for us, it is also interesting to note that um, osteosarcoma, when it develops, is usually, for the most part, effectively treated initially. But then in 50% or so of the patients, it comes back. And when it comes back, it always metastasize, not always, but nearly always metastasize to the lungs. And those lung metastases are very difficult. They're tough. They have survived this onslaught of chemotherapy from the primary therapy, and they're very resistant. But interestingly, the majority of those lung metastases also we found express HER2 because they need the HER2 and some other angiogenic factors to set up housekeeping to become a metastasis in the lungs. And so we use that validated target HER2 to send these T cells that the Listeria has recruited hunting for micrometastases that don't show up yet in the clinic because they're too small, and get the immune system to kill those tumors or those micrometastases before they break out. So HER2 is a validated anti-cancer target. Micrometastases of osteosarcoma are probably, it's definitely more than the majority of them are HER2 expressing. And so that makes them vulnerable in this case. And that's what we use our pet dog Listeria to generate tumor fighting white cells to go after them. And there's really only one system that can interrogate every cell in a person's body and say, is this something that's supposed to be here or something that's not supposed to be here? And that's the immune system. And so that's really the value of this. These micromets of osteosarcoma in the lungs are tough mothers. They have survived every onslaught of chemotherapy and radiation that the patient has had to endure and survive. So they're very resistant to therapy. The only thing that's going to find where they're hiding and eliminate them using a different mechanism of action that they're not resistant to is the immune system. And that's why we're doing this.
Yeah. Thank you, Robert. I'm loving the metaphors today, by the way. I wanted to shift gears a little and discuss the regulatory implications behind OST. Maybe not implications as much as just challenges. I know that you have a couple designations, FDA designations for OST, including orphan, fast track, rare pediatric disease. I was curious, Paul or Robert, how the company are you planning to use or currently using these designations to streamline CMC requirements with regulators?
I'll start, and Robert, you fill in where I miss out on things. But I'll tell you, the opportunities are huge right now from a regulatory standpoint. We did just have our end of phase 2 meeting. The non-clinical and CMC was stuff all that we could handle, and the clinical stuff we're continuing the conversation with the FDA, but it's full dialogue. We've gotten orphan and fast track and rare and pediatric disease. We're going for RMAT and breakthrough designation. We're also going for the Commissioner's National Priority Voucher. What we're hearing from the FDA is very positive. If you have a safe drug, it should be on the market. And we have a very safe drug. And our end of phase 2 meeting last Wednesday with the FDA, there are no concerns around the safety profile. So that's a great start. And then we believe that we have a good response with 35% of our patients. And, we've found that the regulatory process, not just the FDA, but EMA as well as HMRA in the UK, have been very positive and very collaborative. So we're excited about it. Gerald or Robert, anything to add?
Nothing to add. We take advantage of these regulatory designations, so maybe we can have a little bit of a faster path and a facilitated path through the regulatory agencies, and we use them wherever we can.
And, Tyler, we are going for the priority review voucher, and with those things like RMAT and BTD and some of the other designations, we could get approval even faster, but we're very confident we can get the approval by September 30th, 2026.
That's terrific. And what designations am I leaving out? Because I'm sure I'm missing some. Could you list them off for our audience that OST
Sure.
has?
Orphan, fast track, rare and pediatric, RMAT, which is regenerative medicine, advanced therapies, which is of particular interest to many of your listeners. Breakthrough designation, and then the new one is the Commissioner's National Priority Review, which BTD would reduce the review time by about to about 5 months. RMAT would take it to 2 or 3 months, and Commissioner's National Priority Review would take it to 1 or 2 months.
That's great. Thank you, Paul. Not to sound like a broken record here, but again, I really think the whole bacterial vector aspect of OST is it's just really fascinating to me. And I'm curious from a regulatory standpoint if there are any unique regulatory considerations regarding this live vector.
Yeah, of course. So the FDA's, and other regulatory agencies, want to make certain that you're not going to develop a bacterial vector that could create some sort of a super bug if it gets out into the community. And so we have to go through great lengths to prove that our attenuation deletion mutations are not reversible under any circumstance. And so that's one of the things they want to be very careful about. And right after treatment for a few hours, we start antibiotics, which kill the bacteria 4 hours later. But, there's live bacteria in a person for 4 hours. And so, okay, is it present in the body fluids? Can someone else who's not the patient get infected by the same thing? And we've done a lot of work along those lines. And the bottom line is it's very, very highly unlikely, because someone would have to almost get it into their bloodstream in order to get an infection. And even if they do, this is attenuated to the point where it's 4 logs or 1,000 times, well, actually it's 10,000 times weaker than a wild type bacterial Listeria that it was derived from. And so even if someone's infected with it, it's not likely they would have disease from it, because it's easily handled by the immune system. But yeah, there are things that we have to do that are extra, and then environmental concerns is one of them, and that's one of the things that the FDA looks at. Just as if you were working with a modified herpes simplex virus for an oncolytic virus or some other sort of pathogen-based vector system that may somehow find itself out in mixing with the environment.
And it's nice to have Robert, as with the institutional knowledge that he has. When we went to the FDA to do the phase 2B trial in kids 12 to 39, their concern was that we believe the vaccine in the body for up to 72 hours, when Robert had already done a bench trial showing that if antibiotics were given at 4 hours, you'd still have the same immune response, but it would shut down shut down the Listeria. And so that was very helpful and a much safer protocol than predecessors with this technology.
Yeah. I'm curious if, well, first I'm curious to hear maybe a little bit more about some predecessors of this technology specifically, but also of those science safety things that you had to show. Was there anything particularly that was just more difficult than the others? I'm sure we wouldn't be here if the results were negative, but I also am sure was not the lightest lift in the world. So out of those kind of proving out the environmental and the bacterial clearance,
Yeah.
what maybe proved most challenging, if not just the most difficult to work?
So I think Paul touched on it. I mean, Listeria is known to be a bacteria that will contaminate food articles, and it can cause a type of food poisoning in some people. There's either lettuce at McDonald's or Taco Bell or some ice cream that maybe got a Listeria contamination. And so, first of all, there's a big sort of fear factor around that. And the reason that it does that is that Listeria, like a lot of other bacteria, when it gets into an environment, it starts to pulls proteins from around it, and it builds a kind of a crust they call a biofilm. And a biofilm is a problem if it happens inside of a person, because if you build up a thick enough crust, the antibiotics that we're putting in can't get through at high enough concentrations to kill every last bacteria, and you might have some persistence. in the early development of this technology, they were not really thinking about that. And they had a couple of patients where, you know, and they weren't giving antibiotics 4 hours afterwards. They were giving it 3 days afterwards, which was too much time. It allowed the bacteria in certain circumstances to attach to a foreign device like an infusion port or a prosthetic device in a person and create a persistent Listeria sort of infection that had to be treated with a lot of the antibiotics to get through the biofilm cure. So that was probably the most challenging aspect of it. But that's where Paul was referring to the work I did that realized it takes bacteria about 2.5 days, the Listeria bacteria about 2.5 days to build up a biofilm that's thick enough that it can protect it from some antibiotics. And we showed that in the laboratory. And then we also showed that as soon as really an hour or two after exposure, if we then give the antibiotics that early, then what we see is the immune system still got the message. Enough Listeria secreted their payload. It got picked up by the immune system. The same number of T cells were developed, and it had the same anti-cancer activity in the laboratory models that we were using it in. So, the amount of time it takes to get a biofilm is different than the amount of time it takes to get the message delivered. And so we figured out that if we give the antibiotics 4 hours afterwards, we've got all the message delivered, and that's not enough time for it to build up a biofilm to protect itself. And that's really important in osteosarcoma because these children have been through the ringer. They almost all have prosthetic devices, bone reconstruction, bone grafts, plates and screws. Some of them, as they're growing, they have to have prosthetic devices that you can extend as their legs grow longer because of amputations. And so, making certain that the Listeria won't attach to that hardware was really critical. And that's where that work came in handy because now we can be confident that we can give Listeria even to someone who has hardware in their body and not worry about persistent infections because we had no instances of infection of implanted devices, prosthetic devices in the 41 patients that we treated in our osteosarcoma trial, which was the first trial that used that early antibiotics. And so, problem, that was the concern. And now the problem has been solved because we know when these patients are getting Listeria exposure, so we can time the antibiotics such that it won't ever become a problem for them.
Thank you, Robert. Aside from everything you just mentioned, is there anything else that the company did to de-risk OST from a regulatory point of view?
Well, I think
We fired some phenomenal regulatory affairs advisors, including PDUFA fee waivers were applied to. We've applied to the commissioner's national priority voucher. We've got a great team of regulatory affairs as well as statisticians working with us and Robert and his team to make sure that we've got all our bases covered at the regulatory agencies.
Thank you, Paul. Robert, what were you going to say?
Oh, no, I was just going to, Paul gets to say things too sometimes. I have a tendency to talk too much. So,
so this one will be for both of you. Whoever wants to go first. Reflecting on the company's pipeline and the development of OST, what lessons have you learned in process development and manufacturing that might stick out to our audience, and might benefit our audience, rather?
I'm going to let Robert have the last word, but I think what I would say, and it's not as much a process, but persistence, is that this technology was ahead of its time. Robert's ideas, the ideas of Dr. Yvonne Patterson and Dr. Nicola Mason at Penn, the folks that worked on developing this 15 years ago, this technology was certainly ahead of its time. And that may be one of the advantages going to the FDA is that, it's been around for a little bit and they've seen different renditions of it. But they've never really had a full application. And so we've proved the safety. And what I think we're showing is the efficacy with patients in a really, really tough cancer, probably one of the toughest cancers. And if we can be successful in that cancer, then we can pivot to other solid tumors like breast cancer, esophageal cancer. But uh, I'll let Robert finish. But I think it's just persistence and the technology. I think was way ahead of its time.
Thank you, Paul.
Now that's true. The other thing I'll say is that, the process is inherently simple, but technology evolves over time. And so we have a lot more genetic capabilities now than we had, 20 years ago or 15 years ago, even. And a lot of that is translating itself into some of the manufacturing process aspects, more genetic screening and that sort of thing has become a highlight, and more genetic verification of intentional mutations. And we start to look at things over time like genetic drift. If the product stays in storage for too long, do you start to get variations in the DNA that can cause genetic drift? And that's something that we are, from a process perspective, we're controlling for now. I think the other thing that was a real interesting process challenge that we really solved is the lyophilization. Um, as it's prepared, the current product needs a cold chain because you have to keep it frozen. You don't want your bacteria to increase in number while you're waiting to administer it to a patient, because the dose is dependent on the number of bacteria particles you put in. And so lyophilization really does a couple of things. One, it stabilizes the vector as it is, and it also eliminates this cold chain that complicates using it for a drug treatment, and it tremendously improves the stability. You know, our lyo process under accelerated deterioration conditions shows rock solid genetic stability over a year, in the lyophilized state. So that's a real valuable process improvement that we have here. Whereas the old school initial product that was developed back at the university level required minus 70 degrees centigrade, which is, dry ice temperature, just to make sure that the bacteria were going to keep their numbers constant.
That's great. Thank you both. And I know we're getting short on time here, but I wanted to give you both the opportunity to answer our show's hallmark question, which is however narrow or broad you'd like to answer. How do you think we could improve the industry and better biopharma? Paul, why don't we start with you?
Well, first getting approved from the FDA for our treatment. Uh, uh, Better bio treatments means more options, right? I'm not sure I'm answering this question correctly, but uh, you know, we've done the clinical trial to show that this is working in really tough cancer, and we'd like to get approved by the FDA and EMA and HMRA. But what better way to prove processes work than getting a product to a patient? There have been no new treatments in osteosarcoma in over 40 years. And so that's what we're trying to change.
Great. Thank you, Paul.
Yeah, and Tyler, I think I'll build on what Paul was saying. That's really our perspective. You know, we're serving osteosarcoma, which is a rare disease, and it doesn't mean it's any less serious for those patients that have it, but it means it's of a lot less interest to big pharma. And if it's and I believe that rare diseases fall into this crack. The FDA has very stringent criteria about getting a drug approved, and the processes and all of the manufacturing controls, rightfully so, to protect public health. But if you're 14-year-old dying with a rare cancer, you don't really care that they're preventing, a massive problem in people that are getting a heart disease drug. You need your drug now, and you'd be willing to accept more risk to get access to that drug. And I think the FDA is making, and the other regulatory agencies are making overtures to try to make it more accessible. But the hoops that we have to jump through are still very, very high, and the bar was set by the big pharmas of the world. And so for a small company like ours, who's leading with their heart and following with their heads, it's painful for us to see even a month go by, because we know that patients continue to die from this disease, where we think we've got something that can help, but it's just a matter of getting it through the regulatory hurdles to get out there. So that's all the big preamble to say, I wish that we could get drugs to patients with rare diseases more quickly, maybe with a little bit lower bar, because the patients need it, and the risk to public health is going to be minimal because there aren't that many patients with disease.
Thank you both. I really hope that nothing but success comes for OST, and that it can stand as an example and a beacon of positive progress. Paul, Robert, thank you both so much for joining me today. And thank you out there for tuning in to Better Biopharma, the official podcast of Bioprocess Online. We'll see you next time.